Today's topic of lecture is fertilization. So what is the definition of fertilization? Fertilization is the process by which male and female gametes fuse, resulting in formation of a single cell called zygote. It marks the beginning of the embryonic development of an individual. So the parts of the male and female gametes. So before discussing the process of development or the process of the fertilization, you should know the parts of the male and female gametes. Now this is, if you see the sperm, this is the mature sperm, and you can see here that this is the head region of the sperm. Now this head region, it contains a large vesicle known as acrosome, which becomes applied to one pole of nucleus in the form of a cap. So this is present in the form of a cap around the nucleus, and the nucleus and this acrosomal vesicle is present near the cell membrane. You can see here the area occupied by the, now this area occupied by the nucleus and the acrosomal cap, it becomes the head region of the developing spermatozoon. And as I have told you, the acrosomal material contains many hydrolytic enzymes. Now the middle piece, the area containing the mitochondrial sheaths, this is known as middle piece of the sperm. Middle piece of the sperm. Now here lies the mitochondria fused together and they form a sheath in the proximal part of the flagellum. So this is known as the middle piece. The rest is the tail of the sperm, which is formed by the flagellum, which causes movement of the sperm. Now this is the structure of ovum. You can see here that the cells lying outside the ovum are the cells of zona radiata. Cells of zona radiata. So these are the cells of zona radiata, and they radiate around this ovum. Now immediately inside the ovum lies the, immediately adjacent to the corona radiata lies the region of zona pellucida. So you can see here, this is the region of zona pellucida, and then inner to the zona pellucida, between the cell membrane of the ovum and the zona pellucida lies a space which is known as perivitelline space, shown here in the pink. While then lies the cell membrane of the ovum, then this is the cytoplasm of the ovum, and this is the nucleus of the ovum. So now when expelled from the from its follicle, the ovum is carried in the stream of follicular fluid. Back and forth movement of the fimbria. You can see here the distal end of the uterine tube has got finger-like projections. These are the fimbria of the infundibulum of the uterine tube. Sweep the ovum into the lumen of the tube through its abdominal ostium. From the infundibulum, the oocyte passes into the ampullary part of the this uterine tube. The onward movement of the ovum occurs due to beating action of the cilia of the tubal epithelium. Peristaltic contractions of the tubal smooth muscle also help in the onward journey of the ovum, so that the ovulated ovum reaches the ampulla about 30 minutes. Here you can see here it reaches in the ampulla about 30 minutes after its release from the ovary. Now the ovum remains viable and capable of being fertilized for about 18 to 24 hours after its release from the ovary. During this period, it receives oxygen and nutrients from the secretory fluid filling the lumen of the uterine tube. If not fertilized within 24 hours, the oocyte degenerates and disintegrates in the tubal fluid. Now the knowledge of period of viability of the ovum and spermatozoa within the female genital tract has practical implications. The fact that spermatozoa can survive in the female genital tract for about 5 days and the ovum remains viable for only 18 to 24 hours after ovulation. It tells us that the fertilization of an ovum can occur if viable spermatozoa are already present in the female genital tract as a result of coitus that take place a few days before ovulation, or if coitus occurs within 24 hours after ovulation. So what is happening with the sperms in the female reproductive system, that it has to go undergo the process of capacitation, and this is the period of conditioning in the female reproductive tract, and it lasts about 7 hours, and much of it occurs in the uterine tube. During this time, glycoprotein coat and seminal plasma proteins are removed from the plasma membrane that overlies the acrosomal region of the spermatozoa. Now this adding of the glycoprotein coat occurred in the epididymis part of the male reproductive system, and the process was called decapacitation. Now the process of capacitation occurs in the female reproductive system, in which this glycoprotein coat is removed in order to make the sperms able to fertilize the egg. Only capacitated sperms can pass through the corona cells and undergo the acrosome reaction. So out of 200 to 300 million sperms, out of these only 300 to 500 reaches the site of fertilization, and they begin to penetrate through the corona radiata. Now this is the first phase. As I have told you the parts, the sperm has to traverse parts of the oocyte ovulated, which the sperm has to traverse. Now the first layer is the layer of the corona radiata. You can see here, these are the cells of corona radiata. So first phase, phase 1 of the fertilization process of fertilization is penetration of corona radiata. Acrosomal hyaluronidase released by spermatozoa dissolve the intracellular substance present between the cells of the corona radiata. You can see here, this allows the spermatozoa to advance through the corona radiata towards the zona pellucida. So this is the zona pellucida, and these are the various sperms which are getting through. You can see here, this is almost reaching to the zona pellucida. Many spermatozoa succeed in penetrating the corona radiata completely and make contact with the zona pellucida. So here you can see 1 and 2, and this one is also coming close. So many of the sperms, they penetrate the corona radiata and come in contact with the zona pellucida. So second phase is the penetration of zona pellucida and acrosome reaction. Now when the sperms comes in contact with the zona pellucida, you can see here, when the sperms, they come in contact with the zona pellucida, it binds to ZP3 glycoprotein, which serves as human specific sperm receptors. So human specific sperm receptors are present on the zona pellucida, which bind these sperms. This binding induces structural changes in the plasma lemma of the sperm head, which are called acrosome reaction. As a result of this reaction, the sperm plasma lemma fuses with the underlying external acrosomal membrane at several points. The fused membranes break away, resulting in the formation of multiple perforations in the acrosome. Through these perforations, acrosine and other hydrolytic enzymes are released from the acrosome, which digest the zona pellucida and allow the spermatozoon to advance through the zona pellucida towards the cell membrane of the ovum. So you can see here the sperm is advancing through the zona pellucida and in the perivitelline space and then to the cell membrane, plasma membrane of the oocyte. So in this picture you can see the acrosome reaction, in which the plasma membrane of the sperm and the acrosomal membrane, they join each other at multiple points and pores are formed, and through these pores the acrosomal enzymes are released and they act on the they digest the zona pellucida layer. Now the third phase of the fertilization is the fusion of cell membrane of spermatozoa and ovum. Now when the membrane containing the head of the leading spermatozoon comes in contact with the cell membrane of the oocyte, the two membranes fuse with each other. As the cell membrane covering the acrosomal cap of the sperm head breaks down along with the anterior membrane of the acrosomal cap, the actual fusion takes place between the O-lemma and the posterior membrane of the acrosomal cap. So you can see here that the anterior the plasma lemma of the sperm, head of the sperm, and the anterior membrane of the acrosomal vesicle, it dissolves and disappears, and the posterior layer of the acrosomal vesicle and the plasma lemma of the oocyte, they fuse. The common membrane so formed break down to create an opening in the O-lemma for the passage of the sperm contents into the ovum. Okay, the entry of sperm contents and the cortical zona reaction. Through the opening created in the O-lemma, the sperm nucleus, proximal centriole and axial filament of the sperm tail enters the cytoplasm of the oocyte. The sperm cell membrane is left behind along with the mitochondrial sheath and remains of the acrosome. Entry of the sperm contents into the ovum leads to the cortical reaction and zona reaction. So you can see here the contents of the sperm are entering the nucleus, the centriole and the so when the contents enter, two reactions occur. One is the cortical reaction and second one is the zona reaction. So what is cortical reaction? As soon as the sperm contents enter the ovum, the cortical granules of the ovum fuses with the O-lemma and release their contents into the extracellular space around the ovum. The material released from the cortical granules, mainly the hydrolytic enzymes, quickly diffuse into the zona pellucida and initiate the zona reaction. So this was the cortical reaction. The cortical granules are present in the O-lemma and they immediately release their contents after uniting with the cell membrane of the ovum and release their the material inside the cortical granules, that is mainly the hydrolytic enzymes, which quickly diffuses into the zona pellucida, and these hydrolytic enzymes, they initiate the zona reaction. Now what is zona reaction? The hydrolytic enzymes released from the cortical granules, they hydrolyze the ZP3 protein present in the zona pellucida. So they hydrolyze the receptors for the attachment of the further sperms. Once one sperm enter its material, this zona reaction prevents the further entry of other sperm materials. This process called zona reaction renders the zona pellucida impenetrable for all other spermatozoa. Even for those which begin to penetrate the zona and had advanced through it to variable extents. The zona reaction plays an important role in preventing polysomy, that is, fertilization of the ovum by more than one spermatozoon. Polysomy may occur under abnormal circumstances, but it results in formation of a non-viable zygote. So phase 4 is the metabolic activation of the oocyte and completion of second meiotic division. So you can see here the number one process in this phase is the metabolic activity of the ovum increases after the entry of sperm contents. They trigger the ovum into activity. The metabolic rate of ovum increases, and this allows the entry of water and other molecules from the tubal secretions. The protein synthesis in the cytoplasm of ovum it goes up, as these are needed for mitotic divisions of the fertilized ovum. Now entry of the sperm contents also stimulates the oocyte to resume the second meiotic division, and the secondary oocyte completes the second meiotic metaphase and quickly proceeds through the anaphase to form the definitive ovum or ootid, the non-functional second polar body. So you can see here the polar bodies and the ootid. The nucleus of the ovum is now called the female pronucleus. Now this is the female pronucleus. The condensed sperm nucleus detaches from the axial filament of the tail and becomes swollen, and the condensed sperm nucleus is now titled as the male pronucleus. You can see here the male pronucleus. It also swells, and the axial tail filament it becomes fragmented and is absorbed into the oocyte cytoplasm. In human, actual fusion of male and female gametes, male and female pronuclei does not take place. Instead, both pronuclei replicate their DNA and move to come to lie in close proximity to each other. So you can see here, and now instead both pronuclei replicate their DNA and move to come to lie in close proximity to each other. Due to replication of DNA, each of the maternal and paternal chromosomes come to be composed of 2 chromatids, so that now the ovum contains 46 doubled chromosomes. The next event in the process of fertilization is the disappearance of nuclear envelope and of both the pronuclei and formation of mitotic spindles in the cytoplasm of the ovum. So you can see here the mitotic spindles are formed, and the membrane covering the DNA of the pronuclei they disappear. The event of alignment of the maternal and paternal chromosomes on the mitotic spindles marks the end of the process of fertilization. At this stage, the fertilized ovum is referred to as zygote. So it immediately divides. Soon after, each of the double 46 chromosomes splits in the centromere region, and their sister chromatids move to the opposite pole of the cell. A cleavage furrow soon divides the zygote into 2 daughter cells. So this is the cleavage furrow, and this divides the zygote into 2 daughter cells. The mitotic division of the zygote into 2 cells, each containing 46 single chromosomes, is known as the first cleavage division. So what are the results of fertilization? Number 1 is the restoration of the normal diploid number of chromosomes. Union of the male and female gametes, each of which is a haploid cell, produces the diploid zygote having the normal 46 chromosomes. Now determination of the sex of embryo. Number 2 outcome is the determination of sex of the embryo. In the chromosomal sex of an embryo is determined at the time of fertilization by the kind of sex chromosomes carried by the fertilizing sperm. Fertilization of ovum by an X-carrying sperm makes a female embryo having a 46XX genotype. On the other hand, fertilization of the ovum by a Y-carrying sperm produces a male embryo having a 46XY genotype. So, first outcome of the fertilization was restoration of normal diploid number of chromosomes. Second is at the time of fertilization, the chromosomal sex of the embryo is determined. Now, number 3 outcome of the fertilization is the production of genetic diversity. Now, intermingling of the maternal and paternal chromosomes at the occasion of fertilization is an important factor that contributes to genetic diversity in the offspring. Random assortment of maternal and paternal chromosomes at the spindle of the first mitotic division ensures that different children of the same parents differ from each other in genotype and therefore also in the phenotype. Number 4th outcome of this fertilization is the initiation of cleavage. Fertilization initiates development by stimulating the fertilized ovum to undergo a series of rapid mitotic divisions. This initial mitotic segmentation of zygote within the confines of zona pellucida is referred to as cleavage.